Hello everyone, welcome to the new session of Organization of Genetic Material. So in this session we are going to study about the structure of DNA. Okay, so we are having two different types of nucleic acid, that is DNA and RNA. Nucleic acid, that is DNA and RNA, right? So nucleic acid was first isolated by the Swiss biochemist Friedrich. So first time the nucleic acid was isolated by a Swiss biochemist whose name was Friedrich, right? Measured in 1868 from the pus cells. He named nuclein. He named nuclein as a nucleic material. For the nucleic material, he names nuclein, right? The term nucleic acid was first introduced by R. Altman in 1889. So the first time the nucleic material was isolated, right? And it got its name nucleic acid by R. Altman. Right? So nucleic acid are the third most abundant component after water and protein in the cell. So if we are talking about the cell, then we are having nucleus, right? And the cytoplasm. So this cytoplasm is having lots and lots of water material. In the nucleoplasm also there is having water. So the major component of the cell is water as well as protein, right? So the third most abundant component in the cell is the nucleic acid. Okay, deoxyribonucleic acid, DNA. So the first type of nucleic acid, that is DNA, we are going to discuss here. So they are mostly found in the nucleus. So if we are talking about the eukaryotic cell, then it is present in the nucleus. The DNA is present in the nucleus, right? It is present in the nucleus. But if we are talking about the prokaryotes, then the DNA is present in cytoplasm itself. No nucleus is present in the case of prokaryotes. Okay, this we have already discussed in one of the session. Okay, so here we are going to discuss about the eukaryotes, the DNA structure. So nucleus, inside the nucleus, DNA is present in the form of chromatin material, right? And chloroplast and mitochondria, right? They are also having its own DNA. Right? Okay. So what are the features of DNA? If we talk about the features of DNA, then they are made up of nucleotides. These DNA structure is having polarity. They are having polarity, right? 3' 5' phosphodiester bond is present. Complementary base pairing is very, very important. Okay, so these are the different features in the DNA structure. Double helix structure is present, and Chargaff rule. This rule is very, very important to understand how purine and pyrimidine nitrogenous bases are present in the case of DNA. Okay, so we will discuss one by one these different features of DNA. Okay, so first of all, we are going to discuss about the nitrogenous bases in the DNA. So here we are having two different types of nitrogenous bases, that is the purines and pyrimidines, right? Purines and pyrimidines. So pyrimidines are single cyclic structure. Okay, so they are cytosine and thymine. They are present in DNA, right? And it is cytosine is denoted as C and thymine is denoted as T, right? And instead of thymine, uracil is present in the RNA. So here we are discussing about the DNA. So thymine is present in the DNA. So this is the pyrimidine structure. Now if we talk about the purine are two cyclic structures, two cycles you can see in the case of adenine as well as guanine, right? So adenine is represented as A and guanine is represented as G. So these denotion that they are cytosine are represented as C and thymine is represented as T, adenine is represented as A and guanine is represented as G is very important. We will see in one of the polynucleotides, right? Complementary base pairing refers to the specific pairing of the nitrogenous base in the DNA and RNA. So they are showing complementary base pairing. Complementary base pairing. So what is the first of all base pairing and what is the complementary base pairing? Means there are some bases that pair, right? So in the case of adenine always pair with the thymine. So A pair with the thymine. A pairs with the thymine. Cytosine pair with the guanine, right? So C pair with the G. C pair with the G. If we talk about the RNA, then adenine pair with the uracil, right? They are not having thymine. Instead of thymine, they are having uracil. So A pair with the uracil. And cytosine pair with the guanine. So C pair with the G. Okay, and here A pair with the U. So these four nitrogenous bases involve this base pairing. Now complementary base pairing means A is complement to T and T is complement to A. In the same way, C is complement to G and G is complement to C. Okay. So that is why it is called as complementary base pairing. Nucleosides and nucleotides, very, very important. So nucleosides and nucleotides are the fundamental components of the nucleic acid. They play critical roles in the DNA replication, RNA synthesis and protein synthesis. Okay, a nucleoside is composed of two main components, that is the nitrogenous base and the pentose sugar. So if we draw a structure of a pentose sugar like this, then the first carbon, a nitrogenous base is there. So this is the nitrogenous base. This is the ribose sugar, or in the case of DNA, it is deoxyribose sugar. Okay, and here phosphate groups are attached, right? So up to this ribose sugar and nitrogenous bases, this is called nucleoside. This is called nucleoside, right? And when this phosphate group along with the nucleosides are joined, this is called as nucleotide. This is called as nucleotide. So nucleoside do not contain any phosphate group. We have just now seen that if we take out this phosphate group from this DNA structure, then it is called as nucleoside. So nucleoside do not contain any phosphate group. A nucleotide is composed of three components: nitrogenous base, pentose sugar and phosphate group, right? So this is very, very important. Here you can see this is sugar, okay, pentose sugar. This is the nitrogenous base. So this particular structure is called as nucleoside. And when this phosphate groups are attached, to this nucleoside, then it is called as nucleotide. right? So nucleotides are the building blocks of the nucleic acid. Very, very important. So what are the building blocks of the nucleic acid or any DNA or RNA? Then it will be nucleotides, right? They link together through the phosphate group to form the sugar phosphate backbones of the DNA. So we will see this how sugar phosphate backbone forms the DNA structure. Polarity. This is also one of the feature of the DNA, that is the polarity. In DNA, the polarity refers to the directionality or the orientation of the DNA molecule. So if we draw any DNA helix structure like this, okay, so we are having, this is if 3 prime, then this will be 5 prime. And if this is 5 prime, then this will be 3 prime, right? So DNA consists of 2 long chain strands of the nucleotide that run anti-parallel to each other. This is called anti-parallel because 3 prime is here and you can see 5 prime is here, right? And in the other end, we can see 3 prime is here and 5 prime is here. So this is showing anti-parallel structure. Anti-parallel structure. Right? 3 prime to 5 prime, 5 prime, 5 prime to 3 prime, like this. Okay, so one strand run in the 5 prime to 3 prime direction while the other run 3 to 5 prime direction. So this is showing the polarity in the DNA structure. Types of bonds in DNA. Types of bonds in DNA. So we are having 2 very important bonds in the case of DNA structure. Phosphodiester bond. These are strong covalent bonds that link the individual nucleotide together to form the sugar phosphate backbone of the DNA. So this phosphodiester bond. So here you can see 1, 2, 3, 4, 5th carbon is attached. 5th carbon of the sugar is attached with the phosphate. So this is one phosphodiester bond, second phosphodiester bond. So this is called as the phosphodiester linkage. Now they are saying that sugar phosphate backbone. So this particular sugar and this phosphate group unitedly they are building the sugar phosphate backbone of the DNA structure. Okay. So glycosidic bonds now. The N-glycosidic bonds involve the attachment of the nitrogen atom carbon of a sugar molecule. So here the first carbon of the sugar is attached with the nitrogenous bases with the help of glycosidic bond. So these 2 bonds are very, very important for the formation of the nucleotides, and these nucleotides are the building block of the DNA structure. Complementary base pairing is crucial for the structure and the replication of DNA. So here you can see the complementarity in the DNA structure. So A, you can see A is always pairing with the T. Same here. A is always pairing with the T. And T is always pairing with the A. Right? Right? So A is pairing with the T and T is also pairing with the A, right? And you can see here G is pairing with the C. G is pairing with the C and C is pairing with the G. But there is a difference. What is that difference? Adenine pair with the thymine with the help of double hydrogen bond. 2 bonds are there. But in the case of GC or CG bond, we can see C is attached with the guanine with the 3 hydrogen bond, triple hydrogen bond, right? So this is the complementary base pair. which is very crucial for the structure of the DNA, and they are involving these types of hydrogen bond, these numbers of hydrogen bond actually, that is A pair with the thymine with the 2 hydrogen bonds, and C pair with the guanine, that is 3 hydrogen bond. Double helical structure was first elucidated by the James Watson and Francis Crick in 1953. So this double helical structure was first given by the Watson and Crick. So DNA consists of 2 long chain, also called as strands, made up of nucleotides, right? So this nucleotide is very important building block of the DNA structure. The 2 DNA strands run opposite direction, which means they are oriented in an anti-parallel fashion. This we have already discussed. The nitrogenous base of the 2 DNA strands from the hydrogen bond with each other. Okay, so adenine pair with the thymine and cytosine pair with the guanine. So adenine pair with the 2 hydrogen bond and cytosine pair with the guanine with the 3 hydrogen bond, right? This create a spiral structure that resemble a twisted ladder. So here we can have a ladder like structure, but this ladder is not straight, it is twisted. It is twisted like this. It is twisted, right? So this ladder is quite twisted. The structure is often called as a double helix structure of the DNA. So here we can see the double helix structure of the DNA, which is twisted, right? So we are having some peculiarity and some measurement in the DNA structure. The diameter of the DNA molecule has been found to be 20 angstrom. So if we see this structure like this, the diameter of the DNA structure, it is 20 angstrom or 2 nm, right? There is a large major groove and a smaller minor grooves in the double helix. So here we can see this is a larger groove, means at this particular side this space is more, so that is the larger groove. Okay, and there is a smaller groove, also. So larger groove in the duplex, we can see it is about 20 angstrom, 22 angstrom, whereas smaller groove is around 12 angstrom. Okay, the 2-strand coil in the right-handed fashion, each turn of the helix is around 3.4 nm. So here we can see one turn of the coil. One turn of the coil is around 34 angstrom. Okay, one turn of the DNA is around 34 angstrom or 3.4 nm, and they consist 10 nucleotides. So if you count the number 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So 10 nucleotides are present. 10 nucleotides are present in 34 angstrom, that is having one turn of helix, right? And then we can find out the distance between the 2 nucleotide. How can we find that? So we can find in the way, so one turn length is 34 angstrom or 3.4 nm, right? And one turn contain 10 nucleotides. So distance between two nucleotides we can find by dividing 3.4 nm by 10 and getting 0.34 nm. So the distance between the two, nucleotide is 0.34 nm or 3.4 angstrom. If you want to convert into the angstrom, you can write in that way also. So this is very, very important from the examination point of view. Some sort of numerical can be asked. So if you remember, the one helical turn contain the distance and one helical turn contain number of nucleotide, that is 10. So you can find out the distance, and in that way how many turn are present, that can also be find out, right? Okay. Chargaff's rule. Chargaff's rule. This rule allows to determine the percentage of all bases present in the DNA sample if we know any percentage of any one basis that is present. So we are having C, G, A and T. These four nitrogenous bases are present. Okay. Adenine is roughly equal to the amount of thymine because thymine always pair with the A and A always pair with the T. That is, adenine always pair with the thymine and thymine always pair with the adenine, right? So they are roughly equal. Their percentage is equals. Same way, the cytosine C is paired with the guanine and guanine is paired with the cytosine, right? So it shows that the percentage of cytosine and guanine must be equal, right? So this is the basis for the Chargaff's rule, right? So let us see how can we deduce the percentage of three other nitrogenous bases if we know the percentage of one type of nitrogenous base. So let us see with one example. Here, suppose in a DNA, the adenine percentage is 30%. Okay, we just know the percentage. There is a DNA structure. This is a DNA. And in the sample of this DNA, we know the percentage of adenine, that is 30%, right? 30%. Then what will be the percentage of other nitrogenous bases? That means percentage of C, percentage of G and percentage of T. Okay. So we know that A always pair with the T. A always pair with the T. So if A is 30%, then T will also be 30%, right? Roughly equal, that is 30%. So A plus T, what will be the percentage? 60%, right? And if the total sample is suppose 100, then we can subtract this 60. That will be 40%, right? And we know that G always pair with the C. So G plus C, what will be the G plus C? 100% minus 60%. So that will be 40%. So what will be the percentage of G? This is the addition of G plus C. Then we know that G is equal to C. So G will be 40 divided by 2. Though that will be 20%. So percentage of G is 20%, right? And C will also be 20% because G is equal to C. So we just know the percentage of A, that is 30%, and now we can say that C is 20%, G is also 20% and T is 30%, right? So this is all about the structure of DNA. We will have one more session to discuss about the structure of RNA. Till then, thank you very much.